Now it's my great pleasure to formally introduce Ali Ertürk to you. So when you look at Wikipedia, Wikipedia tells you Ali is a neuroscientist, an inventor, and an artist. And I think this is probably the best description of what Ali is doing. And I think he does everything really high level. And I think you will learn a lot about this triage maybe in the next hour or so. And Ali is today the director of the Institute of Tissue Engineering and Regenerative Medicine, ITERM, in Munich, participating in the Excellence Cluster there, and is associated with the Helmholtz and the university, and is an inventor in the best, I think, scientific inventor in the best meaning of the word. Ali has co-invented or invented tissue clearing methods that allow us to gain insights into the 3D morphology of large organisms, and which is crucial to understand biology. Not everybody needs to do molecular biology. We still need to understand the structure. Yeah, we are still far away from understanding the structure and how structural things interact. And I think Ali's work is showing that in an excellent way. He has received numerous awards and has published almost only in the highest ranking journals, I would say. And yeah, we really look forward to your talk, and we really look forward how your work also in all the different fields that you are working in, that you are inventing, and can contribute to cancer neuroscience, can interact with the topic of cancer neuroscience. So Ali, the floor is yours.
Cool. Thank you very much, Frank, for very kind invitation and introduction. It's my pleasure to be here. I was also planning actually since some years to get together with the CASET folks, and I was even trying to do some active emailing. No one cared at that time, but good that now at least there is some interest. I'm happy that I could come together with you guys. So today I will actually talk about some published work, of course, but also some recent work that we just put into bioRxiv. So I'm going to post these links to the bioRxiv papers. If you can download them, it would be quite beneficial for you to follow them. And let's see, I mean, I would be happy to do some brainstormings as well at the end where things go in terms of technology and their applications. So let me start the screen share. Okay. I guess you see my presenter picture, San Francisco Bay Area. So it's good to start with San Francisco. So if, you know, we think about all biomedical developments and, you know, millions of quantifications and graphs and all the parameters affecting human life. And if it everything boils down to one graph, I think it becomes the average life expectancy of human being, which actually has been quite pathetic over several thousands of years, around 35. The major reason is that people died simply prematurely, you know, because of infections, wars, and famine. It's just there was a lot of this premature death, but now, of course, thanks to modern medicine and science, we can avoid this with antibiotics, modern hospitals, and treatment options. Things are clearly going in a very right direction in the last 100 years or so. But also now, but you notice there's this flattening of the curve. So we are reaching to this limit now. Things are not working as great. You know, this is not going up to 120, which is actually the limit, really, like the genome-wise limit of human being. So for now, at least. A major reason what's happening this through the end of this graph, we are not able to tackle complicated diseases. If the reason is complicated, that means several genes involved, several lifestyle factors involved. It's not very easy to solve. There's some examples: heart diseases, cancer, neurodegeneration, diabetics, just to name some of them. So then for me, actually a major endeavor has been what I can do to change this graph in the continuous in the right direction to improve people's life. It's not only about living long, but it's not suffering about these last 5, 10, 20 years of life with cancer treatments, Alzheimer's, and so on. We know that it's horrible to have them. So therefore, I really focus myself to generate technologies that could accelerate biomedical research, that we can obtain a progress, maybe progress of a couple of 100 years in the next 10, 20 years, that we get to solve these complicated diseases. And there are many technologies. You may ask, okay, what are the technologies that might do this acceleration for? I have three criteria for that. That brings me my how. So how are we going to do that in terms of technologies? So by using technologies that are unbiased, high throughput, that is scalable, brings 100, 1,000 times acceleration, and targeted. That means it's not just for everyone, but it's for the specific problem to solve. So, and we have been developing these tissue clearing technologies and putting any technology from engineering, AI, and nanotechnology to achieve this goal. So I will give you examples of that. So let's now maybe move into a bit more concrete biology. You hear a lot about precision medicine, but what is precision medicine? If you ask for all people, you might get different answers. So it boils down to a couple of steps. First, really, there is this huge disease heterogeneity for these complicated diseases. Cancer, to name, of course, one of them. There's, if you think about just lung cancer, you cannot just say it's the same from patient to patient. There might be very different mutations. Even in the same person, there might be hundreds, even thousands of different types of cells mutations happening on the way. So this is a first step to understand this complexity, heterogeneity of diseases, which luckily we have good tools: transcriptomics, proteomics, Organism-Ali, just to name some of them. Problem with these technologies, they are unbiased in application, when we look at the things, but they are biased in the selection. Let's say if you are doing transcriptomics, you have to choose tiny piece of the tissue, then do transcriptomics on that. You ignore all the rest of the organism, where it might be having these different types of cancer cells, so what you didn't sample it. So there's a major problem still lies in this first step, understanding it. Heterogeneity needs a global view, not just picking up tiny things. Second step for me, in personalized medicine, is now we know this heterogeneity, we understand we should develop specific treatments, maybe multiple treatments for this heterogeneity. And the third step, which is also very critical, is to deliver the treatments to the cells of diseases, right? If you don't want to just deliver everything to every cell, we know chemotherapy is not good, but we have to be much precise, one thing, and we have to also achieve the targeting at higher efficiency. If you think about antibodies, for example, for them, it's not easy to get into complicated tissues, into brain, for example, or so. So even there might be targeting, they may not be able to. So we have to make sure all the disease cells, only them, they are targeted. So our technologies, I think, now we are focusing our technologies to solve each problem here, or help solving each problem in these steps. So let's start with understanding disease heterogeneity. As you know, we are an organism of interconnected systems. Head to toe, we are wired with vessels, nerves, and other things. And clearly, these systems, they communicate. If there is a cancer, it uses, of course, the vascular system, lymph system, it moves around. And if you just actually choose an organ, a tissue, and study diseases, we would be ignoring the rest of this interconnected system. ideally, therefore, we have to study diseases at whole organism level. But of course, we are limited. We cannot do this easily. If you think about, you do histology, you cannot do histology of a whole mouse. So it's pretty difficult. So therefore, you have to just choose whatever brain, lung, and section and look at it. If you think about imaging tools, we have some solutions to look at the whole organism, but the issue is that like MRI, PET, or bioluminescence kind of imaging, you might look at whole thing, but we are not looking at this cell level information, molecular level information, which is needed to understand this heterogeneity of the disease. If we want to understand, okay, cells and molecules, disease mechanisms, we can do histology, serial EM. The problem is then we are picking up tiny piece of tissue, nowhere we are looking at the big structure. So our technologies, tissue clearing, tissue transparency, overcomes these problems in terms of imaging, combine these two arms so that we can look at gross anatomy at subcellular level, cost and time efficient way. For example, here, MRI of human kidney with standard method, we can really look at it at cell level using our technologies. So what is tissue clearing? You might have heard of it. If not, let me explain very briefly. For sure, you heard about histology. What we do is standard. We section tissue, stain it, look at under the microscope, which has been great for, of course, decades. But the problem becomes now, again, if you want to look at the big picture, don't want to miss the rest, section is not going to help us. If we are also looking at complicated 3D structures like vessels and nerves, section, again, is not going to help. We will lose this integrated network information. Therefore, we and others have been developing so-called optical tissue transparency technologies where we could convert opaque biological tissue into transparent structure. So you can think this, converting milk into water. Now we can see through and scan through to obtain cellular level information on intact biological specimens. So when I started my lab, well, actually, when I was doing this work, initially my PhD, we did clearing on spinal cord tissue and the mouse brain. And then when I started my lab, I said, okay, let's be fully unbiased, study whole organism. And then we wanted to then make the whole mouse bodies transparent, the project led by Chen and Marika in the lab, where we could initially really render the whole mouse bodies transparent using UDisco technology. But at that time also, we could scan the whole mouse body for the first time with a fluorescent microscope. So that was a big step ahead. And then in 3 years, we developed the new version so-called VDisco, where we could amplify the signal about 100 times. So here, for example, we are looking at Taiwan GFPM mouse, where a subset of neurons are labeled with GFP. So it's a neuron-specific reporter mouse. On the left with UDisco, what you see is some nerves, maybe in the brain and spinal cord, but not much, because there is really high autofluorescence. This signal is not very visible. But when we do amplification about 100 times, the signal on the right-hand side, again, through intact mouse, skin, and bones, becomes really like pops up. You see now every detail. So that was for us like a breakthrough now. We can study a lot of biological things that we want to do. So VDisco truly renders the whole mouse transparent. It's not a joke. So if in the air, it looks like glass, but if you immerse it in a solution, you don't see anything anymore. So this is really like crazy. So then we can use light sheet microscopy technologies to scan now these transparent tissues end to end. So as a neuroscientist, I was very interested in first applying this technology to look at the nervous system of an adult mouse. Again, here, Taiwan GFPM mouse. What we could see is that, nervous system of an adult intact mouse. So that was really cool to see using this reporter. But just also to test the utility of this technology, what we did is actually a local brain injury on the right somatosensory cortex of the mouse. A local brain injury. Now, instead of looking at to the brain only, which normally a standard lab would do, we look at the whole mouse body. And if you focus these axons innervating the shoulder blades, what we see, there is enormous decrease in these axonal innervations. at this axons innervating the shoulder blades, there is this decrease of these innervations compared to controls. This is actually, I think, extremely simple experiment, extremely easy to understand, but it tells a lot. Even a local lesion that you do would not stay local. It spreads. We were not looking, doesn't mean it was not spreading. It was spreading. We didn't notice it. So it actually, to be on the safe side, we have to really look at the whole organism level information. I guess cancer metastasis is a pretty clear example of that. We will come to it. So, of course, we do transparency and imaging of these different systems through the intact mouse using laser scanning microscopy. That means tens of thousands of optical scans and terabytes of data. You know, there is no way that someone can sit and analyze this using ImageJ or eMaris and so on. It's not, they're not going to work. So therefore, we have been developing AI-based tools, particularly deep learning, of course, which is the most powerful for computer vision image analysis. We develop algorithms to analyze cancer metastases and drug targeting. And we develop algorithms to analyze the vascular systems and also algorithms to analyze intact human organs. So if you look at now VESAP technology that we developed a couple of years back, Mike developed a technology where we could now label the whole brain vasculature down to capillary level, some dual labeling of the dyes. And then Johannes developed the algorithms that could segment, deep learning algorithms that could faithfully segment all this imaging data. You can see now here, this is the output of the algorithms. You can see really these large capillaries, vessels on the cerebellum and cortex, but also tiny capillaries now if you dive into the middle part of the brain, all of them are visible. So now we have this extraction of all this vascular information down to a few micrometer capillary level throughout the intact whole mouse brains. After that, what we do is register it to LM Brain Atlas and get information about vascular structures, their density, thickness, branching points, and so on in 1,200 plus brain regions. This is actually, in a way, very exciting. I'm very excited about this technology because vessels, actually, if you think about it, vessels, actually, they're proxy to any pathology. If there's a tiny tumor here, anywhere, this vessel will start to change, they will change their shape. If there's tiny plaque, it will be the same. So what we want to do, use this technology and to identify very early stages of the diseases and then dive into, get them out and then run the molecular analysis that I will show you in the next slides. And also it was one of the papers I posted in the chat, you will see DiscoMS technology, which allows us to go from identification of these initial pathologies to molecular profiling. Now things are becoming really powerful. It's not just now we look at beautiful images, but now we are getting into molecular mechanisms. So looking into vessels in the whole brain, but we were also looking at throughout the whole intact mouse bodies, the heads, vessel systems. And we were interested in also meningeal lymphatic vessels that was also in a way rediscovered some years back now by Kipnis, Karya Alitella, and Mike Niedergar labs. And then we could see now these lymphatic vessels through intact skulls. We don't need to split them anymore, but not only we see them very intact, but we see any cells inside of these vessels. While we were doing these experiments, we actually identified something interesting. So when we look at the cross sections of the mouse and the heads, what we see is that the skull, of course, has enormous amount of immune cells, and we identified these channels that has these vascular connections about 100 micrometer away from the meningeal surface of the brain, and there are these immune cells inside. So that this was very interesting to visualize. And then we named them skull meninges connections, and then we started now on trying to understand their function in brain diseases. that here you can see in this image, because you see in the skull marrow, of course, the bone marrow, there are really hundreds of thousands of immune cells and their progenitors. And if there is any pathology, because these gates are like very close, they can immediately open and then or allow more trafficking of the cells, and they get into brain. And this whole thing gives us a new perspective. Maybe instead of trying to always treat things by delivering to brain, but maybe we don't need to do this. Maybe we can do now. We can use skull to diagnose and treat brain diseases from a new perspective. We have been doing some experiments related to this. This is the other bioarchive paper. I posted in the chat. So you see, for example, here we applied a local stroke lesion on the right-hand side, but we are looking at the whole mouse head on a neutrophils labeling reporter mouse. And you can see this enormous increase of immune cells in the skull marrow compared to controls. So we do a local change, local injury in the brain, and then skull shoots up all the cells. There seems to be a communication in between skull marrow and brain tissue. So that's for us very interesting. And also we identified brain-related proteins in the skull marrow. So we observed the same structures, actually not only in mouse, we studying skull samples from post-mortem human skull samples with meninges. Is the same. We see all these connections in human tissue as well. Then we dive into now more. So we identify this channels. There are, of course, now a lot of questions. I think one important question is skull just another bone marrow, or is it unique to brain, or it has some functions unique to the brain? Second, overall, what is the human relevance? Is there any relevance in human diseases? I think these two key questions to be addressed. And in this study, we did single cell transcriptomics, mass spec proteomics, tissue clearing, compared 6 bones throughout the body of the mouse. And we also did TSP or PET imaging of patients to look at skull and brain at the same time to get information. So in the first study, if we look at, for example, bone marrow cells, using single cell transcriptomics of 100,000 plus cells, we see some heterogeneity. So there's clear heterogeneity among bones, and skull seems to be actually having its own cells located in different locations. But also if you look at the individual genes and their pairings, we see that skull has the most unique ligand and ligand receptor pairs as shown here. And if we move to mass spec, we see more or less similar data. You see heterogeneity, bones are different, some of them clusters, but skull seems to be more different than other bones. And also in PCA analysis, you can see that skull seems to be standing alone among the bones that we studied. And so what is the relevance this time in terms of changes in terms of molecular signatures in brain injuries? For that, we did, again, this MCA or stroke lesion. And what you see now in controls versus in the stroke lesion, there's actually skull becoming even more split, has more unique molecular profile after a local brain injury. We can see the also expression of differential expressions of genes, again, highest number we observe in the skull marrow. Proteomics data shows similar thing. Again, skull marrow is uniquely localized. After MCO, you can see this shifts compared to sham and naive. So then we want to dig a little bit more into the human. So the whole data that I showed, part of it more in the paper, shows that there's clear bone heterogeneity. So when we say bone marrow, maybe we have to say X bone marrow because they are different. So I think that's a very clear conclusion. Second, to understand the human relevance, we already observed the channels, but for me, something was actually quite interesting. Also to see, can I go back? Okay. So these channels, the bone marrow in the skull was very close to the surface. It's ½ meter. This is a human skull. So imagine you're holding your skull. If you take a kind of pin and put, probably you will immediately hit to a bone marrow. This is a bit scary, actually, if you think in a way that you might easily contaminate the immune cell populations into your skull. But the cool thing is, because they're so close to the surface, now you can either manipulate them or you image them if they are interacting with the brain or mirroring. So to understand this, we have been doing this TSPO PET imaging analysis from stroke, MS patients, 2 different groups, and for RT, this is a motor plus dementia-related disease. You can see actually in this crowded images, usually the skull has always high signal. So you see in the brain, but skull always also. So there's a mirroring of the inflammation in the skull wherever it's in the brain. I think that was very crazy. You can see also in the quantifications, brain regions potentially related to disease, they are increased. For example, if you look at the middle graph, it's like MS, 2 type of MS. There's the increase actually is more in the skull base, because you can imagine MS is, of course, the brain part, spinal cord. Inflammation goes down, and we see this immediately in these images. And in dementia, it's more, or in stroke, it's more limited to brain and wherever is the lesion. So the TSPO PET, we didn't choose this randomly, because TSPO PET, when we look at actually the molecular profiles between bones, we observe that in the 5XAD dementia animal Alzheimer's mouse model, you see this TSPO PET protein is actually more in the skull marrow than wild type plus other bones. So there is a specific increase already. So therefore, it makes, of course, sense to look at all this patient's data. We did actually more in-depth study on AD patients. We have here a cohort of 50 plus patients and very clear mirroring of the brain inflammation in the skull regions. So overall, we see that skull actually mirrors the underlying inflammation in the brain diseases. So here we look at into neurodegeneration or stroke, but you can imagine similar things happening, brain cancer related things. So these are things that we are also interested in, happy to discuss later. So what is, in summary, what is the impact and translation of this study? So overall, there is what we observe is there's clear heterogeneity among bones and brain pathologies. If there is something in the brain, it induces changes in the skull specifically. So that's exciting, because now we could potentially use this skull to do some monitoring of the diseases, or maybe even move to manipulations. So I think transferability to the clinic is, I think quite high. In one way, it is exciting that we don't maybe need to deliver now things into brain. Imagine now neuroinflammation, everyone is excited, working on different molecules, but the problem is how you deliver things into brain. You cannot cross the blood-brain barrier, especially if you're doing a large molecule. You want to be specific, but being specific has its cost. Large molecule doesn't cross the blood-brain barrier, but now maybe we don't need to. Maybe we will just do treatment at the skull level. If we use anti-TREM2, maybe antibody, it will just reduce the inflammation in the skull, maybe will have an effect into inflammation. All speculative, just exciting science to think of. So I think there is this extremely cool view, but beyond that, you can see already clinical application there. We could do TSPO PET imaging skull. And here one has to be mentioned. Of course, you can say, well, if you're doing TSPO PET, you look at the brain anyway, why you want to look at the skull? But the cool thing is actually skull inflammation, when we quantify, actually always higher than the brain. So that means actually early stages of the disease, potentially this signal will be only visible at the skull, but not in the brain. So that can bring us really early stages and then detection in terms of diagnosis, and potentially the drug treatments. If there are some treatments, we could also follow them through skull, maybe using some remote sensors. These are also some exciting things. So let's move into some fields that maybe you're more excited. Cancer metastasis. You know that most of the people 90 plus that I learned from the literature might be different compared to different cancer types, but in average 90 plus patients, they die from metastasis, not from primary tumor. And you know how it happens. You know, there's hundreds and thousands of tiny metastases splitting from primary tumor. They get into circulation, blood circulation, lymph circulation. They go, most of them are eliminated by immune cells, and if there's treatment, most of them are eliminated. But even some leftovers, they hide, find niches, and then when the immune system is down, they form the large metastasis and kill the person. Clearly, identification and making sure that these tiny metastases throughout the body are eliminated is absolutely needed, I think, for real treatment of cancer. At this point, we always have some treatments which delays the death, but I think unless we identify all these tiny metastases and make sure that they're not there anymore, there is no security to live really or be cancer-free. So we have to tackle this, but how? I mean, the imaging tools, again, if you look at bioluminescence, MRI, PET, optoacoustic, fluorescent tomography, name them. So all of them will give you a similar thing. To have something, to have a cancer image, you have to have thousands of cells together. Nowhere you're close to visualization of this tiny metastasis. So we said we could overcome this problem. Together with Zeiler Lab, we transplanted memory carcinoma. About 2 months later, we made the mouse transparent, and now we look at mCherry expressing cancer cells in mouse body. One thing, and second, we also treat the mouse with 6A10 antibody developed by Rainer Zeidler here for targeting this cancer specifically as a treatment. So we wanted to see how good is this antibody-based treatment. Standard imaging, you see the primary tumor tree metastasis, not much visible at the liver and lung area. The same animal goes through transparency, light sheet microscopy, plus deep learning analysis. Now you see actually there are hundreds of tumors that were visible before. If you look at now this in 3D, you see this primary tumor close to the leg, but also you see really a lot of tumors throughout the body, in the muscles, even in the bone marrow, of course, in some organs. And a lot of them in this model in the lungs. So you can see really in the lungs down to single cell, even you can resolve the clusters of the cancer metastasis. Not only the metastasis, we can see also therapeutic antibodies or drugs. Here you see when the given antibody is targeting, it is in white. When it is missing the tumor, it is in magenta. So antibody-based drugs, of course, is exciting because they're targeted, they're specific and so on, but how good they're targeting, no one really had real clue about it because we didn't have a tool to look at cell level. I mean, if you don't look at it, it's just a prediction. But now we're looking at this with this technology for this particular antibody, for example, we identify about 20 to 30% of the tumors, they were not targeted. So something has to be clearly improved. Now we cannot just say, this drug is working if you just look at the large tumors, right? It's working. And if we do PET, bioluminescence, that's what we get. We will look at the large tumors. Yeah, this is working. Let's bring it to clinical trials, which will fail because we didn't really tackle this once that has been targeted. We're very invisible. So tissue clearing, I think, quite exciting. I think looking at beautiful images is good, like looking at stars. But if you don't do something about what you see, actually, it's just like, you know, looking, not really acting. So we need to move from seeing visualization of these tiny things to understanding why they are there and what can we do about them. So for this brings me to second step of personalized medicine. So we understood now this is heterogeneity at whole mouse level, but now we have to study the molecular pinning of this tiny metastasis, and we can do this now using discoverance technology that we developed, that we can isolate them, you know, from these regions and then run unbiased mass spec-based proteomics. So this is quite powerful because, you know, identification of this tiny metastasis variable and then getting them out and, you know, going into thousands of proteins, which related is this. Now we have a tool to compare the ones targeted, untargeted, small ones, big ones, different organs. Enormous amount of mechanistic biology is extractable. So you might say, well, tissue clearing is a process of chemistry, chemicals, you do dehydration, organic solvents. How do you get the protein out? Actually, that was a good question for us as well. When we look at actually using the disco methods that we use, we obtain like the protein amounts similar to fresh tissue, highly reproducible all the time. We said there's something wrong here. This cannot be correct. Yeah, so we were not convinced that it is high quality. It cannot be. We repeated again. We repeated different instruments all the time, the same results. We are very happy that it's not only mouse but also in human tissue archived for several years. We could clear, image, and then run proteome, obtain similar reproducibility. I'm very, very actually excited about this. You know, I cannot really say how it is for me enormous step ahead in a whole process. So when look at the protein groups, actually what we see that there is not a shift because of clearing. All these things might remove some of the proteins. Maybe there would be bias. Only bias we see actually is related to blood proteins. You see actually there is less blood proteins in VDisco. A major reason is that because for clearing, of course, we wash the blood out compared to fresh tissue. That's actually even just a positive control that is expected. So, and then you can look at all the protein groups that relate to the different pathways. You see no shift, so it means you can study them faithfully. So also protein groups that are related to or subcellular structures, organelles, they are also highly preserved. For us, the most unbelievable thing again was the membrane, because it has the most lipid, and we do clearing using lipid extraction. And again, here we were like, yeah, this cannot be true, but somehow it's true. And I'm happy to discuss the reasons. I have some ideas on that. So we then applied this to some studies, some this biology. We did this traumatic brain injury on microglia animals where there's EGFP expression, and now you see in magenta. So this is now standard experiments. You do something, a manipulation, clearing, imaging, now AI, identification of this inflammatory regions. Now we have like a path of microglia. We can take out some small regions and study now region by region this heterogeneity of microglia, and identify the heterogeneity of inflammation at different regions. That was quite also interesting for us to get into this heterogeneity of disease in the biological tissue. Of course, we use deep learning to analyze all this complicated data to identify the plaques, for example, in Alzheimer's model. So you see on the left, actually, maybe it's very hard to see, there are some red signals that are identified by using this technology. Here is better highlighted. You can see them. We found this very few plaques and take them out and then run proteome and obtain again heterogeneity of these plaques throughout the brain. So basically our technology, if you think about, people are very excited, of course, now spatial omics, right? So it's now the new hype thing. But when people say spatial omics, actually it's not spatial. It's a bit of deceiving term, because it's a tissue slice. Tissue slice is 2D. There's no 3D, so it's not spatial, it's 2D. So, but true 3D comes from something like this. We have the whole thing image. From there, we can move into omics. Then it becomes really omics. So in our discoveries overall brings the technology of the spatial omics in terms of imaging of volume to full scale. It becomes also makes it unbiased at the imaging. So, there is not anymore if you section the brain into 1,000 sections and okay, which one I have to now do spatial omics. You have to pick. Now you don't have to, you just image, and now where is the interesting thing, you can take it and run your study. So the summary of the discoveries you can see now in this mouse brain, where 6 weeks old Alzheimer's mouse model. These initial plaques, they weren't reported, no one knew there are some plaques here. Using again clearing and imaging, we are able to look at cell level, the whole mouse, and deep learning identification of these really initial plaques. We can take them out using some new technology we developed and run mass spec proteomics and obtain 2,000 proteins at least from these tiny plaque regions. So this is, I think, very exciting. Again, combining these two technologies, now we're looking at early stages of diseases. I think that's the key here. We know a lot of diseases we have to tackle at early stages. We have to understand their early stages of molecular underpinning, and now we have a shot at this. So now let's come to the third stage of precision medicine, where now we identify maybe some molecular pathways to these tiny metastases. Now, we develop a molecule, small molecule, let's call it. We want to now deliver them to the cancer cells. How are we going to do that? So we have been, of course, clearly, if you use small molecule, it's not targeted. If you use antibody, is targeted, but it has problems that it cannot cross a lot of membranes. And nanotechnology is the way to solve this problem. And I guess with the lipid nanoparticles and vaccines, now it's clearly proved itself in clinical use. But we have been working on more fancy nanoparticles. Those are DNA nanorobots with Henrik Dietz lab, because DNA origami, DNA, what you can do is actually you can, like Lego bricks, you can fold them and create different structures. Lipid particle is a like a what? Like a balloon. It's the same. You won't be able to change its shape, but like this, you can make different structures. You can make really complicated structures. You can see some of them here. So you can do scissors that can, punch the membrane. You can make carriers of small molecules, large molecules, all these crazy things. So cool thing is that they're biological, so they are from DNA oligos, so therefore they are degraded, so there's not side effect, and they are programmable for multifunctions. You can do different things. Let's say find the blood-brain barrier, open the blood-brain barrier, get into it, find the microglia, destroy it, or so, all these things can be coded into these robots. But again, when people are trying to develop these nanotechnologies, there's one of them, others as well, they use technologies like path imaging to study them, right? So we deal with nanostructures and we use imaging that works at millimeter scale. So there's like 6 orders of magnitude difference. Yeah, so this is just disconnected. So we cannot again study here targeting at cell level, at least what we need in terms of resolution. You can imagine now using our technologies, we can do that. We have been using now a DNA nanorods here. And then what we did actually is to conjugate them with antibodies to be targeting here to specific cells. We then, this is CX3CR1 for immune cell targeting. You can see very nicely targeting in the liver and in the immune organs lymph nodes. If we have now the origami structures that is not targeting, you see they're mostly taken up in the liver as expected and secreted. You can even see throughout the parts. But now you can also add antibody that is targeting to cancer cells. Now you see it's not anymore in the organs, but in this throughout this tiny metastasis. So now here the one example that is for immune cell targeting, you can see the targeting of origami in, of course, most of the immune organs, and clearly a lot of signal in spleen has I don't know, some millions of immune cells. And very clearly we can see clusters of immune cells, but also individual cells if we zoom in. In addition to that, I don't know what is this black lines now. So you can see, for example, if you move to different organs, in the liver, again, individual immune cells are being targeted. A nice thing, actually, not only you see at cell level, if you look at the sections, optical sections, you can see at subcellular level, like puncta, that means they're taken up into the cell, actually. And then now we move to the lungs, you can also see very nice labeling of the immune cells, individual immune cells in the lungs. They're being targeted as this antibody throughout the lung structures. So now we are able to target immune cells and potentially any cells that we are interested in using this technology. So I have been showing you a lot of data from mouse. You might ask, okay, we work with human tissue and so on. Can we apply this system to human tissue? So, well, we wanted to do that. At the beginning, we had a lot of problems, but then Sean solved this problem by developing a method called Channel. The problem was that these detergents that we use for permeabilization, they didn't work well for started human tissue because they were large. Our screen identified chops with small miscellaneous size, could permeabilize the tissue end to end, render whole human organs like human kidney transparent. You can see not only transparency, but we could do labeling of the structures, the capillaries and Organism-Ali and so on, and we look at them in high detail. So we are able to now really generate cell level visualizations of immune organs. So here, deep dive into it, you can see Organism-Ali, and you can really see cell level resolution around Organism-Ali. So now we want to use this information to engineer organoids and human tissues. And we know that a problem with organoids, the core is dead. There is no good vascularization, but we have now vascular map of human organs, which then can be used as a blueprint to create, for example, a kidney organoid, which faithfully really reflects real human kidney, and then we can print homogeneous structure of human kidneys in small ones in multi-wells to do high throughput drug screen, but also we can use the whole map of a human organ and move from there, generating intact human organs for transplantations. So a bit futuristic, maybe it's going to take 10 to 20 years to go there, but we will be there. I think when we are there, the whole medicine will be different. So with that, I would like to end, and I'm happy to acknowledge the team, AI team, great students, robotics team. I didn't show much of their stuff, but also we have all this multidisciplines and multinational working together happily. And I'm happy to take your questions.
Yeah, thanks a lot, Ali. That was gorgeous, highly inspiring, I must say. So, of course, in archaeology and medicine, we know that the first surgeries that are documented in humans are trepanations, so removal of the skull of people. I think 5,000 years ago that started. And there was always the question, where did people that 5,000 years ago removing parts of the skull? So maybe they knew better than us today. Maybe they knew how to treat certain diseases by doing that. This just came to my mind. And when I was writing, I had the pleasure to write an editorial for Nature some years ago, where Dorothy Sipkins and her team, they described this funny bone marrow brain channels that they found in the mouse, and I was skeptical to the end. And I said, okay, I just want to see this in humans. I'm not sure that this will ever exist in humans. And then I saw your paper, and I was just thrilled that this is not just some funny artifact, but really something that is also possibly, because clearly the bone marrow is so important for cancer, right? I mean, circulating cancer cells reside in the bone marrow. And Dorothy has shown in her team that leukemia cells use this road to enter the brain and to make a meningeal disease in this. And it's, of course, a big question whether other cancers are using this route, right? Whether everything is just circulating cancer cells in the blood and then extravasation. It could be that a lot of cancer cell gets access to the CNS by that. So I think that's all fantastic. I have 1,000 questions, maybe one, because then I need to stop. Or maybe 1.5. So first question is, do you see also changes in other bone marrow regions in the spine, or in the extremities, arms and legs, or is that really restricted to the skull?
A good question. Actually, in the bioarchive paper, again, you will see actually there are information back to it. I think yeah, please go ahead, do maybe a journal club and give us feedback. So this is what I ask you. It would be good. Bioarchive papers needs feedback actually from experts like you. Yes, what we see, it's actually there's shift. If we do brain injury, all the bones, they change, but skull shifts more than the others. And skull and vertebra shifts similar to each other than the others. So I think there is this domain-specific duties and immune cells being populated. A lot of biological information there to be extracted. We just see that this there are still changes in other distal bones, but their purpose probably is different than what is maybe they do increase the immune cell population for whole body defense or against other things. But in the skull, you see this increases related to neutrophils that travels through channels and to the meninges. We see the same population. So there is this brain specific change in the bone marrow of skull.
Great, thank you. And one very short question. So coming to your multi-omics, which is also, of course, fascinating. I mean, now, how do you locate these tiny spots in the cleared set or the cleared sample and then transfer it to proteomics? How do you, I mean, what is the technology that it enables you to do that?
So when we do clearing, we do imaging. Of course, we then this identifies in the light microscopy, we can see all these tiny metastases or plaques. And then you can then take them out using different technologies. Or simply we did in this paper, in the bioarchive paper, you can section it, laser capture, and then run to the proteome. So that's easiest to do. There are a bit more complicated things to do. I can tell later maybe about them.
Great, and I would also love to learn, but maybe we can make this session how you employ AI and deep learning to make sense of all these huge data sets. But maybe somebody else wants to comment on that. And
sure,
I've already.
So maybe we start with some questions from the chat, Frank. Or...
Yeah,
yes, sure.
You want to read, or people?
Yes, I simply maybe read it.
Okay.
Do you want to? So you can.
Yeah, if they want to ask, they can ask. If not, you can read it. Yeah.
All right. So I think we have some question from Pius Yoshi, multiple questions actually. So asking whether the residual tumor particles that you showed, whether they cause tumor recurrence in the mouse or whether they are just residual tumors which might not really be oncogenic anymore.
Mm-hmm. So this is unknown for us at this point. But we know that tiny dormant tumors can become active later. There are many evidences of that. So basically, it's a lottery. I would not play lottery. I would bet. I would try to eliminate all of them. So I wouldn't just say, "Oh, they are probably not going to do anything." So, but at this point, we don't know what is the percentage, what are the mechanisms to attempt to triggering them to become again active. Yeah.
Then I think there's a more technical question, but I think this is also very interesting for the larger audience from Amruta Vatak, who's asking, this is which clearing method you would recommend if you simply have mouse organs fixed in PFA or cryofixed to visualize whole organ architecture. She's working on mouse cancer models where she would like to adapt the method.
Yeah, so we know we develop so-called disco clearing methods, but there's other methods like cubic, like clarity, you may know. I objectively, very objectively, I can say that disco one is the best one. And but we wrote this manuscript review. It's called what is it called? A guidebook for disco clearing. It's in molecular systems biology. Please go and read that. It's step by step, tells you which sample, which staining, which clearing has to be used. Just there's like a chart that you walk through that can help you. But overall, disco clearings, they I think prove themselves to be most consistently working in every setting.
Yes, thanks for this very nice literature advice. And another question, again, also referring to the clearing methods, and Veratil is asking, I'm wondering, how do you cope with the different organ structures during the clearing process? Like bones need to be decalcified, while other organs don't. So
yeah, how many TB data? Okay, one mouse data could be 2 terabytes. So how big is the data she's asking? Maybe it could be a few terabytes to 10, 20 terabytes, really depend on the resolution we scan, but it's in the order of some terabytes. So we use at whole mass level, we use default decalcification and decolorization. That means removing the red color of the blood. But what we don't see really much, decalcification doesn't really affect much these other organs. Probably it affects, but it's really minor. We don't see any disturbances in the structure. So overall, we don't really have any problem there using the global decalcification and looking at different organs.
Okay. Great. Then a more biological question from Jeremy Borniger, asking whether you have to worry about eliciting a host immune response to the DNA nanorobots.
Good point. So what we do when we design nanorobots, you can choose your oligos, and we know all the pathogens, DNA, viruses, and so on. We double check, and that should not reflect. And we know also what kind of DNA is trigger immune response. Regardless, still this is foreign DNA. It can produce some immune response. It did actually. But what we observed, this is really like we did all these experiments, FACS, etc., etc. They're really like minor things, and usually it comes back after a shot. I guess if you do like if you get vaccine, probably whole immune system goes crazy and then comes back. Something like this. If you take something or hit your hand, there is some immune response. It's something like this. I think it will be like that. If we develop DNA origami-based pills, you take it, maybe a bit shift of immune response, but it's going to quickly come back to normal.
Okay, great. Then one last question from the chat is from Ines Quesada, who's asking whether there's a strategy to preserve nerves before clearing and whether it is possible to do immunofluorescence for disco-cleared tissue.
Okay, so I'm trying to see whether there's something underneath of the question I don't understand. Because we already preserve the nerves and image them in VDisco, you can see them very nicely. We have the videos of whole mass nervous system. I don't know what does Ines asking here, what we should preserve.
So I think that was more general question about the technique and whether you would destroy any structures.
Maybe which technique you would choose to label nerves.
So how do we label? We use typically reporter animals and then look at them. So you cannot really do like whole mass level antibody labeling if you're interested in, but we can use the GFP reporter that expresses in nerves. We use this. If you do organs, organ by organ, you have more chances to do antibody kind of labeling.
Okay, great. It's 6 o'clock on point, I guess. So
any other questions live, or maybe one last question. So I just raised this shortly. So regarding the analysis tools, this is certainly something that is crucial, right, to make sense of the data. And you showed pipelines, or you showed some examples of pipelines, how you do it. Maybe you can comment on, because I mean, if that should go into the width, into let's say into many labs worldwide, or you would need, of course, some open access solutions, right, to analyze this data. Is there any ideas to do that, or is your lab already doing that, providing open access, let's say, data analysis tools, or is that still something very local and lab based?
So all the algorithms we develop, we put them in so-called GitHub, where the things are stored and the data is available. But these are codes. Still, they work on this specific platforms. So a biologist won't be able to just take it and use it unless there's some training. So that's a bottleneck. I see that, and therefore I was asking already some years back to companies who develop like Imaris kind of softwares to integrate this kind of deep learning algorithms that people can use. but they could never get into this. I think it's for them a lot of R&D that they are not able to do it easily. And of course, another problem is all this, deep learning. When you develop a deep learning algorithm, usually it's specific for a purpose. So for neuron is different than for vessel, is different than for cancer. And then what you should, there should be more generalized algorithms. We are also working on this. So we want to do a little bit more, some packages that can be accessible by others using some GUIs. But I think my suggestion at this point right now would be to be in communication with your local experts, computer science group, where I'm sure there are groups that do computer vision, image analysis. They can quickly help you. I mean, for them, to integrate these things will be relatively easy. So instead of trying to set up years of work in the lab, just at least start like this with the local experts.
Yeah, excellent. Thank you. Thank you, Ali. Maybe, and so if you have some time to stay a little bit longer with us, and we say goodbye to all guests. Yeah, we see a lot of clapping hands. I think I only join the clapping hands here.
Thanks.
And yes, thank you. So I think we meet again in 2 weeks from now with a working seminar, and you get the invitation, or you got the invitation with it. So see you again. Bye-bye.